So our next step in the genetics section is to talk about mutations and how mutations can lead to genetic disease, genetic disorders. Also important to note too, mutations are not always bad things. Sometimes we tend to focus on the negative parts of mutations, but things like us being able to walk upright, changes in position of organisms' eyes, evolution is caused by mutations. Now, some of the bad ones, we've talked a little bit about cystic fibrosis before. This is just an example here. So typically, inside our respiratory system, we have mucus that lines the respiratory system. And this acts as our first line of defense to trap viruses and foreign particles from entering our respiratory system, infecting our respiratory cells and causing things like chest infections, pneumonia, etc. Now, the problem with cystic fibrosis is instead of the cells that line the respiratory system producing sort of a thin mucus that traps those particles, it gets really thick and sort of oozy. Think the last time you had a chest infection, bronchitis or something, and you were coughing a lot and you were coughing up, big chunks of phlegm. That would be similar to what somebody with cystic fibrosis would experience. So at some point in time, a gene that would have been in cells, in the gametes of one of our ancestors, would have had a mutation, a change in the code that instead of it coding to make a thin mucus, coded now for making a thick mucus. And because it was in a gamete, a sex cell, that got passed along to children, or a child. That child's cells all had that mistake in the code, and now that child produced thick mucus, and their gametes had that same gene in it, and then they were able to pass that gene on to their children. So that's a problem when we look at mutations. If a mutation happens in a body cell, that only affects the organism or the individual that mutation happened in. If it's a mutation that happens in a gamete, a sex cell, then it passes along and continues to be passed along through that, through the family history or the family tree. Now, a mutation, heritable change in the sequence of DNA. So this is something that can be inherited, can be passed on. And it's important to know a lot of these mutations happen naturally. Okay? Your DNA is replicating so quickly for some cells, especially things like skin cells, that because there's so much code to copy, trillions and trillions of base pairs, that the copying of those base pairs, even if a cell was 99 point, a whole bunch of 9% effective at copying DNA code, there's going to be the odd mistake. So this happens naturally. But other things like ultraviolet rays, X-rays, gamma rays, certain chemicals that are in the air, the food, the water, those things can all increase the chances of these mutations happening. Now this section, or this bit of DNA on the board here, might represent a section of our DNA. So we've got AAC, CAG, TAT. Now there are different types of mutations that can happen. We can get a point mutation, or we can get what's called a frame shift mutation. Now a point mutation would be something like this. If we were to look at each one of these codes, each one of these codes has a specific amino acid name that goes along with it. So from our data book, we can look up AAC and see that this stands for asparagine. CAG, when we look that one up, stands for glutamine, and TAT stands for tyrosine. So those are the amino acids that DNA sequence would stand for. Now, if there was a point mutation, what could happen is maybe some radiation, maybe just random mutation that happens when we're copying the DNA. Maybe we get a substitution mutation, where that A is now substituted with a C. Okay? Now the problem is ACC, when we look that up in our data book, does not stand for the same amino acid. It now stands for ACC, stands for threonine. So a totally different amino acid has been produced. These amino acids stay the same because those codes didn't change, but this first one, because of that one nucleotide that was substituted, now we get a different amino acid that's produced. Now, if we go back to what we had before, again with the original AAC, a different type of mutation that can happen is if we, instead of substituting a nucleotide, we would insert a nucleotide in between or into the code, or we could delete a nucleotide from the existing code. So we can do an addition or a deletion mutation. So let's look at an example where we do an addition mutation. Let's in between the C and the A in this part here, let's insert another cytosine. So what's going to happen is that cytosine is going to get inserted in here, in between those, and then everything is going to shift. And we call this a frame shift mutation. So instead of us reading CAG, TAT, everything is going to slide down a little bit, and we're going to have CCA for this part. Okay, CCA. The G is going to go into this next code. So G, T, A, and then that T would be going with whatever sequence was over here. Okay? So when we read the code, AAC still codes for the same amino acid, but now we're not reading CAG, we're reading the new code here, CCA, and CCA codes for proline, not glutamine, codes for proline, and GTA codes for valine, not tyrosine. So again, this was the code, and we got this amino acid sequence. Now by inserting a C in here, okay, a little arrow showing the C going in there, everything shifted down one nucleotide to the right, and we got a different sequence that produced a different amino acid sequence. Now again, going back to our original code, the other one I said could happen is we could get a deletion mutation where one of these letters gets removed. One of these nucleotides is completely removed. So let's say this cytosine right here, that cytosine, when it's copied, when the DNA code or DNA strand is copied, replicated, that C gets missed. So we're going to take that C out of there. And then again, just like before where everything shifted one letter to the right, now everything is going to shift one letter to the left. Because remember, we need to read the DNA code in triplet, in 3-letter codes. So AAC, oops, AAC is that one. Then, because the C bumped over here, the next code starts AGT. And then the next code is AT something, whatever would have been off in that position over there. So just for simplicity here, let's say there was a C there. Then that C that would have been the beginning of this next code is now going to become the end of that code. Now when we read this, AAC, well, AAC, hey, we had that here before. We had the C before we removed it. So guess what? We're going to get the same amino acid produced. We're still going to get the ASP. Okay. Then for the next one, now we're reading AGT. AGT codes for serine. Okay. Different than what we had initially. Now we have a different amino acid there. And ATC, let me look that one up, that's isoleucine. It's not tyrosine, we get isoleucine. So there we have the definitions for those two mutations. We have a point mutation, which was just where we substituted one letter, one nucleotide for another. And then we had a frame shift mutation where we either removed or we added a nucleotide between existing nucleotides in the sequence, and that caused the entire codon list to shift one letter down. Now it's important to note whether it's one amino acid that's out of line or has been changed, or a whole series of amino acids that have changed, it's going to have the same effect. If the protein, or if the amino acid sequence isn't correct, the protein isn't going to function. If one amino acid is wrong, the protein isn't going to function. If it's 13 amino acids that are wrong, the protein that isn't going to function. I use the analogy of like your locker combination. If you had, let's say you had a locker combination that had 7 numbers that you needed to type in or you needed to rotate through, right? So you need to go to the right to get to the first number, the left to get to the second. You needed to do 7 of those different numbers instead of the typical 3. Well, if one of those numbers was off, the locker won't open. If 6 of those numbers are off, the locker won't open. So the same idea, if one amino acid is out of order, then the protein doesn't function. If 16 amino acids are not correct, then the protein won't function. Now, just some terminology, things called mutagens. These are going to be things that can increase your chances of mutations happening. So this could be, again, food, could be environmental factors, things like ultraviolet radiation, etc. Carcinogens would be mutagens that strictly or simply cause cancer. So just some terminology to be aware of. Now, something that we look at in Science 30 still is to look at pedigree charts. So this is a page taken out of your data book with some symbols on it, and it's almost like a family tree, but the family tree is typically used to indicate or to show patterns of inheritance of genetic disease typically, but we can use it even to look at things like brown hair color or curly hair color. But typically we do pedigree charts to look at disease and see how that's passed through a family. Now, some symbols here. We'll have the female as a circle, the male as a square. Individuals that are affected by the trait are shaded in, and then a line in between 2 individuals would show that they're producing children. So up to this example here, this would be a mom and a dad. This would be, if this is, let's say, you here, if you were a male, then that would be you. If you were a female, you would be a circle down here. That would be your dad. That would be your mom. This would be your uncle. That would be your aunt. And we typically go in order of birth. So if this is your dad, that is your aunt, that's your uncle. Your uncle was firstborn, your aunt was secondborn, your dad is thirdborn, and then this is your mom from a different family. Okay, she would have her own pedigree chart over on the side, and then this is you affected by whatever the trait is. Now, this is just an example of a pedigree with cystic fibrosis, and it will tell you, in Science 30, will tell you whether it's autosomal dominant or recessive. That would be where you do the big A, little a, that type of thing. Or if it's sex-linked, where you'd have to put in the X and the Y chromosomes. So for example, with this one here, what we might expect you to do from something like this, since it says it's autosomal recessive, what I would do, I'm going to use the C to indicate the trait, okay, or to indicate the allele. So that would be for normal, okay, not having cystic fibrosis. And then a little c, that would be for somebody who had cystic fibrosis. Okay? Now when we look at the trait, because it's autosomal recessive, that means these individuals here have got the disease. So since they've got the disease, they both have got to be little c, right? That's the only way to get the trait. Now, all of these other individuals in this family don't have the disease, so not having the disease means that they've all got to have at least one dominant big C. Okay? At this point, we don't know if they're big C, or heterozygous big C, little c. All we know is they don't have the disease, so they have to have one big C. But we can use this to determine what these other parents would have. So for example, this kid right here, this boy, who's affected by cystic fibrosis, little c. Well, one of those little c's have to come from each parent. The other little c had to come from the other parent. So that tells us right away these parents have got to be heterozygous. There's no way for either of these parents to be big C, because they wouldn't then produce a child that was little c. Okay? Now, if we go up to this person here, that's, grandma. First generation, this would be grandma if this is you, one of you down here. Okay? She's got a little c. So that means one of her little c's has got to go to, and we already figured that out here, each one of her children. So that has to be a little c. That has to be a little c. That has to be a little c. Has to be. There's no way we could have that happen any other way, because Dad, even though he was a big C, right, gave a big C to each one of the children here. The only thing Mom had to give was a little c. Now, for the other ones, now it's kind of up to question a little bit. We're unsure whether Dad is a big C, or a big C, big C. We can maybe assume that he's big C, big C, because none of their children came out as little c, little c. But, as we learned with probability, there's no guarantee that if he was a big C, little c, that any of these children would have it. There would be a 50% chance, you could do the Punnett square and figure it out, if this was a little c, that 50% of these would be little c, a little c, they would have the disease. But again, just like boys and girls, you can have a family with 8 kids and all of them are boys or all of them are girls, even though it's a 50-50 chance each time that you're going to have that trait. Okay. We can get into other ones where we have autosomal dominant. So Huntington's disease, autosomal dominant. And in this case, if we do the same thing, we would use maybe the big H, okay? Because it's dominant, that means you have, Huntington's. I'm just going to keep it easy, Huntington's. And if they have the little h, that would be for normal. Okay, so now what we need to do, normal individuals, we're going to do this sort of backwards. The ones that aren't affected, these have all got to be 2 little h's. Okay, has to be all little h's. And that's a little different than what we just did with the autosomal recessive disorder from the last question, where the individuals that were shaded in wound up having the autosomal recessive trait, or sorry, the homozygous recessive allele combination. But now we're looking at autosomal dominant, which means you get the trait by having the dominant allele. So this individual here got the trait, got to have at least one big H, one big H. Again, kind of like the last example, we don't know whether they're big H, or big H, little h at this point. We just know that they have one big H. But what we're going to be able to do again is use their children to figure out what that other allele is. So for example, grandpa back here, he's a big H, grandma, little h, little h. If you look at their children, they have a child here, a child here, a child here that are both little h, little h. So that means one of them came from one parent, the other one came from the other parent. Grandma here has 2 little h's. The big H came into this one from the dad, same with this one, same with this one. The only thing the grandma has to give are little h's. So guess what? Little h, little h, little h. And this person down here, this female who has Huntington's disease, big H, little h there. That's a little messy. That should be a little h for that one right there. So little h went into that kid right there. Now there are a number of these pedigree charts in your textbook and on worksheet provided. Good idea to go through those and to also try a sex-linked trait. Actually, you know what? Let's do a sex-linked one next. Now this is a pedigree chart for a sex-linked trait, and in Science 30 again, we would tell you that the individual, that the trait was passed along using sex-linked inheritance. So from this example, there's a few different characteristics or a few different symbols on here. And in Science 30, we wouldn't typically tell you or wouldn't typically use symbols like this in Science 30 to show a carrier. So that would be a hint that this is a sex-linked trait as well. We would tell you in Science 30 that this was sex-linked. But what you have to do now is use your X's and your Y's in here. So there's Queen Victoria, for example. Here's Prince Albert. Okay, and they had some children. Now, in this example here, Albert doesn't have hemophilia. Hemophilia is a recessive disorder, it would tell you. So since he doesn't have hemophilia, you would have to put in the capital H up there. Since this is showing Victoria as we call carrier, she doesn't have hemophilia, so there's the big H, but she's a carrier for hemophilia. So we'd put in the little h. Now they had some children. Leopold was one of their children, X, Y, right? Because it's a male who's affected. And since they're affected, we have to now put a little h up there. Okay? And then we could go through the whole family here. Another example is we've got Tsar Nicholas II of Russia and Alexandra, who was a granddaughter of Victoria. Alexandra, X, doesn't have the disease, but is a carrier of the disease. And Nicholas II, male, X, Y, doesn't have the disease. Okay, so that's how we do one of these that's a sex-linked. Now, as I said, a sex-linked disorder, they will show you that it is sex-linked. They will tell you that it is sex-linked in Science 30, so there's no guesswork. Typically, with a sex-linked disease, you would see more males affected with the disease than females. You may not see any females affected by the disease, meaning showing the disease. These females, okay, sure, are affected, meaning that they're carriers of the disease and they could pass it on to their children, but nobody in this family that had hemophilia is female, and that's typically one way you would tell that it was sex-linked.